on average, 70 beats per minute, per hour, per day, per week, per month, per year. Whew. I know I can't do that much work. Having the setup of the membrane reduces the friction and lets the heart beat smoother. People who have issues with that membrane or the fluid surrounding it, it's very painful. They actually feel every beat of their heart and usually end up in the hospital having to maybe get injections, have stuff pulled up, whatever the case might be. So membranes are extremely important to the parts of our body. The membranes are usually named for the specific cavity or organ in which they're found. For example, pericardium. Peri means around, card refers to the heart. So pericardium is the membrane around the heart. Pleura is the lungs and the thoracic cavity. Peritoneum is the abdominopelvic cavity. So the membranes are usually named for what they're close to. Now, how do we know a lot of this material? X-rays, imaging, doing dissections, looking at different things that could have happened, such as in a car wreck or something like that. But we have learned so much about the body from imaging techniques since roughly the mid-1900s, because our ability to image something has improved greatly. So we have stuff like radiography, which is just an X-ray. All right. In the beginning, when they had this come out, they were not aware of some of the dangers that come with it. But now we are. Okay. But we've also increased our abilities. We kind of mentioned the ultrasound on Monday. CT scans, which used to be very primitive and used to take a long time, but now CT scans have gotten really good and gotten quick. You know, I was like, whoa, they finished that CT scan already? We have some other types of imaging techniques that probably you may or may not have heard of, but that's because they're so specific, okay? They're very specific as to what they do. Angiography, for example, is blood vessels, okay? Spatial reconstruction, giving like a 3D view of reconstructing bones, for example. MRIs, which have come a long way. PET scans. Anybody in here known anybody that's had a PET scan? A PET scan? Mm-hmm. Of what? Of the brain. Usually. PET scans are, they're usually, that technology is very unique, okay? And PET scans are usually the last option because of, it is very expensive to get them done, the technique is very unique and that sort of thing, but we learn a lot from PET scans, a lot. This would be an example of just an X-ray. The goal or the job of X-ray is to simply show bones. It does not show us any soft tissue, only the hardened tissue. If we look at ultrasound, and I do believe the 4D ultrasounds have gotten better than this. All right, I think now you actually see the baby's face even clearer than that. CT scans, these right here, they can pretty much do a CT scan of anything. You know, head, abdomen, pelvic, pretty much do a CT scan of anything. CT scans show us soft tissue. They not only give us a view of that hard tissue, but the soft tissue in and around that hardened tissue. The reconstruction, like I said, it's like a 3D image working. Usually you see it in relationship to like bones, for example. This is that angiography, which is the blood vessels, showing the blood flow, which I find amazing, okay? MRIs, the job of the MRI, soft tissue, cartilage. Those are the jobs of MRI. And then PET scans. Note what it uses. The PET scans use radioactive labeled glucose. Now let me kind of give you an idea of why I'm kind of pointing that out. What is the smallest molecule that our cells can use? Glucose. And the only source of energy for brain tissue is glucose. Brain tissue cannot use any other food source. The only thing it can use is glucose, which is why if your blood sugar gets low, you get foggy, like you can't really think straight, you feel like you're off balance and that sort of thing. It's because the brain is not getting the glucose it needs. So one of the things that the PET scans show us a lot about is activity in the brain, because the radioactivity of that glucose molecule lights up. So areas of the brain that are active show up, but areas that are not don't. Do you happen to know a disease that this imaging has helped tremendously?
Schizophrenia.
I think so. Alzheimer's. We have learned more about Alzheimer's with PET scans because, like I said, it's usually the last option. It is a little more used now than it was probably 10 years ago, all right? And they have found that there are certain areas of the brain that don't light up for people with dementia and Alzheimer's. And it has allowed them to better create medications to, there's no stopping Alzheimer's, okay? But they have been able to delay the progression of it. And PET scans have been a huge help in doing that. So we have learned tons of information from using PET scans. This is just a list of all of them together in one spot. Now, we need to look at this last little bit on your study guide. It will come in a little more in Chapter 2, but I want us to do Lab 1 today. And so I've got this on here because of Lab 1. This is, of course, something that hopefully goes back to your biology course. But look at what our questions are. What is a gradient? How does it apply? What is flow? How does it apply? What is a pressure gradient, concentration gradient, electrical gradient, thermal gradient? Now, Lab 1 that we'll do after we get back from break, and we'll talk a little more about it then, but Lab 1, it's kind of a mixture of information, okay? They used to have them separated out into 2 different labs, but they kind of put them together. And so the first part of Lab 1 is about scientific notation, okay? Using the metric system, all right? The other part of the lab, which is like you're doing tons of like chemicals and all that sort of stuff. So rather than actually exposing ourselves to all these chemicals in the lab, we're going to kind of like read them, look at them, that sort of thing. But what it's referring to, yesterday we kind of determined we're in human anatomy and physiology, so therefore we're kind of dealing with the human cell. And the animal cell has this beautiful thing called the plasma membrane. Does everybody remember that from general bio? And we have inside of it that nucleus, all right? Got that endoplasmic reticulum, we've got the Golgi apparatus, all that sort of stuff. The plasma membrane. Does anybody remember the structure of the plasma membrane?
It's like a double-sided kind of thing with a hydrophilic head and hydrophobic tails that allow for passage of like certain molecules into cells.
Okay. It's a lipid, that phospholipid bilayer, all right, where we've got those phosphate heads, the lipid tails, and the way that they arrange themselves, they arrange into a bilayer, a double layer. So in looking at this structure, it's the perfect environment for cells. We have the outside of the cell and the inside of the cell. The outside is a fluid environment. The inside is a fluid environment. Embedded into it is a whole bunch of proteins. Okay? On the outside, you could have tons of materials out there. You could have ions. You could have amino acids. You've got all this stuff on the outside, and when the cell needs something on the inside, it works to get that material. All right? Now, in looking at this information, when we say a gradient, that means materials have to flow. When we have a gradient, with a gradient, materials tend to flow. Notice it tend to, okay? From an area of high concentration to low concentration. Does everybody remember that? So, if we had a high concentration of a particular ion on this side of the cell, if it can flow into the cell, it has now flown or moved with its